Hi children. So in the previous session we discussed about carbon dioxide is necessary for photosynthesis in plants. So in this session I am going to explain about light and photosynthesis. So in the previous session we discussed about air and water and photosynthesis. Air and water, these are the two factors that are mainly involved in the process of photosynthesis, and how they are involved in the process of photosynthesis already we discussed and proved by some scientists. Okay, so in this session, so the light and photosynthesis. Light is also very important factor for the conducting of photosynthesis by the plants. Okay, here in the Priestley's time, so the scientists did not understand about energy. So whenever the plants are synthesizing the food material, that food material is released the energy for the growth and development of the plants. So as well as the plant food is taken by any animals, so the animals are also need energy for their growth and development. So how the energy is released? So in the plants and animals. So in the Priestley's time, scientists did not about it. So but later and much was discovered about it, how the energy is released from the food material. Okay, so the energy is released from the food material. So when the carbon dioxide and water is formed, so by the combining or combination of the oxygen with the carbon and hydrogen, so when oxygen combined with the carbon dioxide is formed. When the oxygen is combined with that hydrogen, water is formed. So then what about the reverse? So which means, so the carbon dioxide react with that carbon dioxide react with that water. So what will be happen? So here in the next slide, so just you see that a Dutch scientist Jan Ingenhousz, so in the era of 1730 to the 1799, so he found that answer. Okay, so he studied the way which plants formed the oxygen. Okay, so in the year of 1779, he noticed that it happened only in the presence of light. So the oxygen is formed in the presence of light. That was proved by Jan Ingenhousz, scientist, in the year of 1779. So that's why, so the light is very essential for the formation of oxygen. So in his experiment, so he used aquatic plants like Hydrilla or Elodia. So these two are submerged suspended aquatic plants. So submerged suspended means the plants which are completely contact with the water. Okay, so that's why they are called submerged suspended aquatic plants. Okay, so he observed the Hydrilla and Elodia plants in a bright sunlight. So what he observed? So when he observed in a bright sunlight, so the small bubbles are formed around the green parts of this Hydrilla plant and Elodia plant. Okay, so but while in the dark period, about in the dark condition, so the green bubbles or small bubbles are not formed nearby the green parts. So what does it indicates? So in the sunlight, small bubbles are formed around the green parts. So in the dark light, so they didn't form any bubbles. So after that, it is confirmed by that Engelmann, scientist in the early 20th century. So he detected the point of maximum rate of photosynthesis. So maximum rate of photosynthesis, the light is essential for that process of photosynthesis. So the which light, okay, it is used for the maximum rate of photosynthesis, that was confirmed by the Engelmann, scientist in the early of 20th century. Okay, so how is confirmed the which light is used for the production of maximum rate of photosynthesis? Okay, so just you see here, so he used a alga. Alga is an aquatic plant. So when we see any river or any lake, small lakes or small pond, on the surface of that river and lake and etc. So the green patches, a green thread-like colored moss is appeared on the surface of that lakes. Okay, so that nothing but it is called as an aquatic alga. Okay, so already you studied about a study of microorganisms in your 8th standard. Okay, so remind it one what is alga. So alga is a chlorophyllous Autotrophic, green, thread-like moss that are formed especially on the surface of the water. Okay, so when these algae export to the different colors of light, so already you know that one. So the light is mainly divided into the visible light and second one is an invisible light. Okay, the visible light which is an VIBGR. So what are the colors? So that are 7 colors, that is VIBGR, already you know it. Okay, so he exposed the alga to the different colors of light, so from the violet to the red. Okay, and he used oxygen sensitivity bacteria also. Okay, that found, so they crowd around areas illuminated with red and blue rays of light. Okay, so actually they are illuminating, so especially the oxygen sensitive bacteria that are illuminating the area, so within a red and blue rays of color. So by the conducting of this small experiment, when the alga exposed to the red and blue light, so it show maximum rate of the photosynthesis, so which means, so the red and blue rays are red and blue light, so which is used mainly for the process of photosynthesis that takes place by the plants. Okay, so this led to more studies on effect of light on photosynthesis. Okay, already you know about the different colored compounds which are called pigments. So the plants are shows mainly green color leaves. So some of the plants are also shows yellow colored, blue colored, red colored, orange colored, etc, etc. Okay, so these colored compounds which are called pigments in plants. Okay, so these pigments in plants, that maybe it is a chlorophyll or xanthophyll, yellow color, carotenes, orange color, and etc, etc. Okay, so these all pigments are utilize the light energy, Okay, so these are, so here the colored compounds are called as pigments. That pigments are utilize the light energy. So now oxygen is producing during the photosynthesis in the presence of light experiment. Okay, so here the aim of the experiment, oxygen is produced during photosynthesis in the presence of light. Okay, so it is an aim of that experiment. So you see the picture. So the next step for the experiment is material required. So what is the material required here? One is in a test tube, and second one is in a beaker, and third one is in a water, and fourth one is in a funnel, and fifth one is in a hydrilla plant. So instead of hydrilla plants, we also used Elodia plant. Okay, so for the setup of this experiment, so we follow this procedure. Okay, what is the procedure is, take in a beaker and fill it with a water. So you also some add potassium bicarbonate to it. Okay, calcium bicarbonate or potassium bicarbonate, because of, so the plants needs the bicarbonates, the carbon dioxide in the form of bicarbonates. So that's why you add some amount of any bicarbonate solution to the water. So take in a beaker, fill it with the bicarbonate water. Okay, so then you take in a test tube and also funnel. Okay, so you insert the funnel into the test tube. So in the funnel where the broad opening is there, so in the broad opening you insert hydrilla or Elodia plant. And so this setup was invertedly inserted into the beaker. And this experimental setup was placed in sunlight for 1 or 2 days. So what will be formed? So in the presence of light, the hydrilla or Elodia plant involved in the photosynthesis process. So when it is involved in the photosynthesis process, so it release gas bubbles. So that gas bubbles that are light in weight, that's why those gas bubbles are reached to the top of the test tube. So where the empty space is there, in that empty space, so the oxygen molecules are reached. Okay, So it is an experimental procedure. So here the observation means the plant's hydrilla that releases the gas bubbles. These gas bubbles that indicates producing of oxygen during the process of photosynthesis. So which is in a hydrilla plant. And now you see that second one is in a Elodia plant. Okay, so both are hydrilla and Elodia, so which are submerged suspended species. Okay, so that are mainly involved in the process of photosynthesis and release the bubbles. So now you watch this video. Okay, photosynthesis demonstration. Okay, oxygen produced during the process of photosynthesis. What are the materials required? So the Elodia or Hydrilla aquatic plant, and second one is test tube, third one is beaker, and fourth one is sodium bicarbonate solution, and sixth one light source, either it is in a sunlight or normal light we used in our room. Okay, now coming to the procedure. Yeah, now you take a beaker filled with a water. So in that water, what you take? Bicarbonate solution. Okay, so make a sodium bicarbonate solutions by the adding baking soda with the 3 cups of water. So fill the beaker 2 by 3. Okay, now it is a sodium bicarbonate solution. So you take sodium bicarbonate solutions and that was poured into a beaker. Okay, so after that you also pour some amount of bicarbonate solution in the test tube. Bicarbonate solution in the test tube. So now you insert. Now you take in a funnel or directly you insert Elodia or Hydrilla plants into the test tube. Now you close the test tube with your thumb and invertedly insert into the beaker and arrange in a light source, that is a normal light that present in your room or a light which is under the sunlight. Okay, in the day 1, day 2, day 3, what will be formed automatically? So here the gas bubbles are formed around the plant and also as well as the tip of the test tube, so which indicates the plants which involves in the photosynthesis in the presence of light. So the oxygen releasing during the photosynthesis in the presence of light only. So if we keep this experiment as usually in the dark, so there is no oxygen produced. Okay, so now coming to the next slide. You are coming to the next slide. So what it is? The next experiment is sunlight is necessary to form the starch in green plants. Okay, so already we conduct experiments. So the sunlight is necessary for the releasing of oxygen. So during the photosynthesis that already discussed just now. Okay, so now I'm going to discuss about the sunlight is necessary to form starch in green plants. Okay, for this purpose, so we need what material? So we need a potted plant, so a healthy potted plant. We also need a black paper. Okay, black paper, and we also take the screen gauge. Okay, the screen gauge apparatus is also present in our lab. So either we use the black paper or we use the screen gauge apparatus. So along with these potted plants and black paper, so we also need the material, so which are used for the conducting of starch experiment. That nothing but it is an methylated spirit, iodine, Petri plate, Bunsen burner, asbestos gauge, and etc. that we will discuss in that experiment. Okay, so now coming to the procedure. So first you take in a potted plant that was placed into the dark condition. So why the plant placed into the dark conditions? Already we discussed in the CO2 experiment. So because of de-starching the plant. So for the 2 to 3 days, so we keep this healthy potted plant in a dark room. So after that, so we take the plant and take in a black paper and cut into S shape. So here we cut the S shape, not only the S shape. So as you wish, so you can cut it as an A shape or X shape or star shape or round shape, whatever it may be. So after the cutting of this black paper, so the black paper was faced on both surfaces, but nothing but it is an adaxial and abaxial surfaces. That means lower and upper surfaces of the leaf, and fixed it with the clips. So just you see here, so fixed it with the clips. Okay, so after the fixing, so you have to move this potted plant into the sunlight. Okay, so when the fall onto the sunlight is fall onto the surface of the leaf, what will be happen? So it involves in the process of photosynthesis. So after that 1 or 2 days, so you take the leaf. From the potted plants and conduct the iodine test. So just you see here. So in this big what will be? So the blue coloration is appear where the black paper is not present. Okay, just you see here. So in the S shape there is no black paper, and the leaf apical region and leaf basal region is also not having black paper. Only the black paper is present in the mid area of that leaf. So that's why when we put this plant into the sunlight, what will be happen? So these green colored leaf area, so that receive the sunlight and involves in the photosynthesis and prepare the starch. So where the black paper is present, so under the black paper, the plant tissues are not receive the sunlight. So that's why they do not perform the photosynthesis. So if they perform the photosynthesis, what will be formed? The starch is formed. So where the starch is formed, the leaf is exposed to the sunlight area that only form the starch. So the leaf which is not exposed to the sunlight area is not formed the starch. So that is confirmed with the help of this experiment. So just you see here the black paper is present. That's why the leaf is not exposed to the sunlight. That's why there is no starch. That's why there is no blue coloration. So where the blue color is present, where the leaf that shows or that emit with that light, so shows with that light. Okay, that is only perform the photosynthesis and prepare the starch. Okay, now you see this Ganang's photo experiment. Okay, so here we prove that sunlight is necessary for the process of photosynthesis. Okay, for which is also called as Ganang's light screen experiment. I already told you we take the paper and also light screen of screen gauge. Okay, light is essential for photosynthesis. So first we take in a potted plant. Okay, we kept the plant under dark condition for 2 or 3 days for the de-starching. Okay, so dark treated plant is called de-starched plant. What it is called? De-starched plants. So now you take that. Here we take the screen. Okay, so it is a clip of the instrument that are fixed on that surface of the both adaxial and abaxial surfaces. So when this setup is transferred to that sunlight, what will be happen? This is close to the lower hollow cylindrical box like structure. Okay, here the shape is star shaped. Okay, when it is exposed to the sunlight, so the plants involved in the photosynthesis. Okay, so this light screen is fixed to a leaf of the de-starched potted plant. Okay, so both the surfaces are that are fixed with that apparatus. Okay, so in the sunlight for the 4 to 6 hours, so we take that leaf and place it into a test tube and add our alcohol. Already we discussed in the starch experiment. So boil it. So the plant leaf that decolorized and add in a few amount of iodine drops. So where that light is exposed on the surface of the light, that shows the blue color and remaining that shows the star shaped color. Okay, so this is an experiment. The sunlight is necessary for the process of photosynthesis. So now you write the questions. The explain the moles of leaf experiment with neat labeled diagram. Okay, and the second one, describe the experimental procedure and observation of oxygen is produced during photosynthesis in the presence of light. Okay, third one, how do you prove the sunlight is necessary for form starch in green plant? Okay, dear students, write these 3 questions in your notebook. Thank you. So we will meet on next session.